All right. Second to the last lecture, renal physiology. Primordial sea, oceans of nephrology, just to take a pee. Okay. Urine. The study of and fascination with urine has been part of the medical canon for thousands of years. What was this stuff that we had to do? Why was urine not the same color as the water we drank as it came out? So 400 years BC, Hippocrates, who we see here on the left, was the first to really codify uroscopy in the West by describing the bubbles that appear on the surface of urine, and they can, in urinalysis, that is analyzing urine, can be a way to diagnose various types of kidney disease and other chronic illness. Galen was the first person, and Hippocrates, when I say kidney disease, he didn't call it kidney disease because he didn't quite understand that urine was being produced by the kidneys. When we say kidney disease, he means that there was some sort of disease that was related to the urinary system that was indicated in the quality of the urine that was being passed. It was Galen in the West who identified the kidneys as being the organs responsible for production of urine by taking living apes, clamping their ureters, and watching the kidneys swell, thus concluding that the kidneys were the organs that produce urine. Certainly wouldn't have passed muster today in any research lab, but it advanced science back then. This whole crop of maybe charlatans arose in throughout the Middle Ages into the late Middle Ages of uroscopists who believed that they could diagnose a whole host of physical and metaphysical, often, problems with you just by looking at your urine and the bad humors that may have been in your body because of the quality of your urine. This guy, Dr. Thomas Bryan, spoke out firmly against these charlatans in his pamphlet called The Piss Prophet, which I found to be kind of funny, wherein are newly discovered the old fallacies, deceits, and juggling of the pisspot science. I can't even read. Used by all those, whether quacks and empirics or other methodological physicians, who pretend knowledge of diseases by the urine in giving judgment of the same. So he goes into this long diatribe about basically a mythbuster calling out these quacks. But that is not to say that there is not a lot to be learned by urinalysis. Later, this gentleman here, 200 years later, Richard Bright, a Scottish man at the University of Edinburgh, was the first to identify the correlation between kidney disease, kidney failure, and what he called dropsy. We call edema. This kidney failure can lead to swelling of the extremities, and also in the protein analysis, proteinuria. This glomerulonephritis was known for a long time as Bright's disease because he was the first to identify this. All right, so 20th century, they began to experiment with kidney transplants. And kidney transplant, it was one of the first to be explored because of the relative ease of transplanting a kidney. The plumbing that the kidney requires is very straightforward. There's not really any kind of nervous innervation in the kidney. There's not any nerves that is a deal breaker if they're severed going to the kidney. There's really just the blood supply, which is easy. These big renal arteries, we just stitch them up, and the ureter, you just stitch that up. So surgically, very doable, and they're pretty easily accessed too by an incision in the back wall. So a very good candidate for the first transplant. However, it was first attempted in 1909 using animal kidneys. This did not work. This didn't end well for the person. The body's immune system, the recipient's immune system, immediately recognized the kidneys as being foreign objects and rejected those kidneys. And we had tissue necrosis. It wasn't until 1954 that this guy down here, Joseph Murray, this happy physician, and his collaborators right here in our neighborhood in Boston performed the first actually successful kidney transplant between identical twins, between monozygotic twins, two people whose genetics were the same. And this was done, they didn't require any steroid hormones or other immune suppressants because the recipient's immune system did not recognize that tissue as being foreign because it had the same genetic composition as the host's own kidneys. And these were the Herrick twins. So here we see the twins. These two people who were the first people to undergo successful organ transplant in the world as they inspect an early incarnation of a dialysis machine. Dialysis is a machine that draws your blood out of your body, filters it, and then pumps it back into your body, doing a very poor approximation of what your kidney actually does. So that was a major landmark. We could go on in that vein, but we'll leave the history off there. So what does the urinary system do? Right? It excretes wastes, and then it is responsible for storage and elimination of that fluid waste. And then, quite importantly, it's responsible for a host of homeostatic functions, such as regulating the plasma volume in your blood. This is going to have direct impact on your blood pressure. They do this through the production of two hormones, erythropoietin and renin. Renin is called a hormone. We've been calling it a hormone, but it's actually not really a hormone. Renin does not have any receptor per se. It is an enzyme that causes the cleavage and activation of angiotensinogen to angiotensin and a whole cascade of other activities after that. The kidneys are responsible for the homeostatic regulation of various solute concentrations like calcium or via calcitriol, the production of calcitriol. It can stabilize the pH of your body by monitoring and modulating the hydronium ion and the carbonate ion concentration. And then it detoxifies the blood. If you have some sort of chemical in your blood that the body deems excretable that needs to be removed, and if that's a fluid-soluble, liquid-soluble compound, it's often the kidney that is responsible for mobilization. All right, so just to remind you of where the kidneys sit, just tucked up under the last two floating ribs, easily accessible surgically through the body wall right here. Not really anything major that you would need to hurt in accessing it surgically. Pretty thin body wall here, just a little bit of a floating rib you need to push out of the way. And then it's located retroperitoneally. So this light blue layer here is parietal peritoneum, lining the back wall of the abdomen, covering just a little bit of that kidney and helping to pin it to that back wall of the abdominal cavity. Oh, yes, how fun. Let's do this. Just so that we can have an easy time talking about the physiology here. Jack. Right there, what is that one? Easy. Urethra. The ureter, yeah. Tanner. What is this little structure here? Looks like a tiny hill, renal papilla. This broad region here that all this stuff is coming through, plus the renal vessels, that's called what? The hilum. Yep. And then what is this portion? So the ureter this is flowing into the ureter. What? Renal, like the pelvis, the renal pelvis. That's it. Well, we know that's fat. We can do something harder. Here. Well, what is this? I guess we'll just go up here. What is this void space called? A renal sinus. And I'm not actually sure what this is trying to indicate. But let's assume that it's indicating these vessels here. What are these blood vessels, Liza? So there's an artery and a vein. These are the ones, just for academic sake, we'll say these are the ones that are mirroring these structures here before they enter the actual tissue. So, that would be just up here. It's a tricky question. So these arteries are mirroring these segments, so we call them the segmental arteries. Okay. Is that? Are you looking ahead? Is that what the chart is trying to say? I'm not sure what they're indicating. Well, okay. Okay. Well, whatever. Anyways, James, what is this region, broad region called here? The medulla. Robin, what's this stuff out here? The cortex. Alex, what is this? Just the pink region here called? The pyramid. Okay. And then one of these little branches here, which is what this is indicating. What are those called, Ms. Burlington? Or Burlington? Calx, yeah. Calices, plural. Calx, singular. I guess that's what that's indicating. So then one of these big ones, a major calx. Is this indicating vasculature? I'm going to assume it is. Kelsey, what's the vasculature that's running right along here?
The interlobar arteries and veins.
Veins, yeah, okay. And then Callie, what's this dark covering out here?
Fibrous caps.
Yep, yeah, the renal capsule, fibrous capsule, sure. You two back there. What's left for you? What's the vasculature called that's running along the outside of this pyramid?
Arcuate.
Yep, the arcuate. And then what about the little arteries and veins that are coming off of that towards the...
It starts with C.
Yeah, does anyone want to help her? It's in the cortex, yeah. It's cortical, it is cortical, but they're radiated. They're radiating out into the cortex. The cortical radiate arteries and veins. All right. So we just went through all that blood supply. The kidneys deal with about a fifth to a quarter of the blood supply in your cardiac output. So that's a huge, between the liver and the kidneys, it's almost half of the blood supply. Half of your cardiac output is going through one of those two organs. It's a lot. It's about 1,200 milliliters of blood per minute just sitting there. All right. So we saw this picture in the lab. We have the cortical radiate arteries coming up, going to the afferent arterioles, which go into the glomerulus, and then the blood comes out via the efferent arterioles. The filtration happens in the glomerulus, and then there's this series of tubules that proceed from the glomerulus, the renal corpuscle, into the other parts of the nephron. So let's look. There are two types of nephrons. We said this in lab, the cortical and the juxtamedullary nephrons. The vast majority of them are cortical nephrons, and about 15% are juxtamedullary. This should make sense to you because one of the renal lobes is roughly pyramidally shaped, right? And so we have room for a lot of these cortical nephrons out here, and less room as they converge towards the apex of that pyramid for the juxtamedullary nephrons. But you can pack many more of the cortical nephrons up here at the top. All right. So here's an actual nephron. And we see the first portion is this renal corpuscle that has the afferent arteriole coming in, going into the glomerulus, where we have filtration, efferent arteriole coming out. Those two arterioles are entering the capsule on what's called the vascular pole. On the other end of the renal corpuscle is called the urinary pole, where the filtrate is emerging and entering this series of tubes. The first tube is called the proximal convoluted tubule. And what happens here is during filtration, we're sort of throwing the baby out with the bathwater. We're filtering our blood, and anything that's small enough to pass through the pores, which is all this water, plus really small solutes like salt, glucose, amino acids, anything really tiny is able to pass out with the filtrate. Some of that stuff we don't actually want to get rid of. Like maybe we don't want to lose that much water. And we probably don't want to lose all our glucose. We probably don't want to lose all of our proteins. We probably don't want to lose all of our electrolytes, right? So we do want to lose the urea. We do want to lose whatever toxins may have been filtered out. So what we're going to try to do here first is to pull back in the things we don't want to lose, all right? And that's the job of the proximal convoluted tubule. We resorb water, we resorb the ions that we wish to reserve, and all of the organic nutrients like the glucose, the proteins, and any small lipids that may have passed out through the filtration process. The filtrate then goes into the descending loop, this descending limb or arm of the loop of Henle. You'll notice that there are thin and thick portions. The descending limb is primarily thin. The ascending limb of the loop of Henle, the initial portion of it is thin, and then halfway up it becomes thick again. Right? So there's this thin portion of the ascending loop, and then it becomes thick. We'll find out in a few slides why it looks like this. This mechanism, this loop mechanism, uses something called countercurrent multiplication to concentrate our urine, and or the filtrate. It's not quite urine at this point, and it's pretty ingenious how it works. We'll see how that system works in a bit. After the nephron loop. So, yeah, I don't know if I pointed out that the purpose here is to resorb more water on one side and to resorb salt on the other. So we pull in water here and we form this concentration gradient. This concentration gradient in the tissue out here drives the second half in the ascending loop where we're trying to pull solutes back out of the filtrate. So the concentration gradient that we establish along this axis is going to help us drive the solute gradient, or vice versa. Next is the distal convoluted tubule. This, the action of the distal convoluted tubule is more variable. It's easier to control the specifics of what's happening there. So, and this is where we're trying to get rid of things, active excretion, active secretion, where this is the removal of things from the blood that did not get filtered out, that we still want to actively put into our urine to remove from the body. So this is where we would eliminate various toxins, any kind of acids or drugs or other, say we have too much of a particular ion that we're trying to eliminate, like calcium, for example, although that would be rare. This would happen in the DCT. Next, the filtrate enters the collecting ducts, and we fine-tune the filtrate at this point it's called tubular fluid. We fine-tune it by any final resorption of water and concentration, or resorption of any last electrolytes or other small molecules that the kidneys deem necessary. Finally, through the papillary duct to be eliminated. So let's look at each of these portions in a little more detail. Here's the renal corpuscle. Wow, that's a cool picture. Somebody must have somehow peeled the Bowman's capsule off of a renal corpuscle and then taken an SEM of it. So you can see how you have the afferent arteriole coming in there in the bottom right-hand corner, and then it breaks up immediately into this complex knot of capillaries. And we'll see what it does in a moment. This is really small. It's only about 250, 200 micrometers in diameter. That's quite small. So to give you a sense of that, if you think about the thickness of your fingernail, the thickness of your fingernail is about 1,500 micrometers. So 200 micrometers would be 1/6, 1/7 of the thickness of your fingernail. Very, very small structure. Here is a diagram of the renal corpuscle where we see the vascular pole on the left and the urinary pole on the right. We have the afferent arteriole coming in to the glomerulus, and then the efferent arteriole coming out, a little bit smaller in diameter here in this cartoon, indicating that we have lost some volume. We have lost some volume there as the efflux from the corpuscle. So as that glomerulus goes into this renal corpuscle, those capillaries get coated with these particular kinds of cells called podocytes. These podocytes are going to form a layer of epithelium, a visceral layer, meaning it's right on the organ of the glomerulus. All right. So think of that glomerulus as an organ, and there is a layer of epithelium called podocytes that are covering that. Those podocytes then, right around this point here at the vascular pole, these podocytes then peel off into another parietal layer of epithelial cells. So there's this space in here, this capsular space surrounding the capillaries. This capillary space communicates with this tube that's heading out this way here. All right. So proximal convoluted tubule, glomerular capsule right here. This is the parietal layer. These podocytes on the glomerulus here, these podocytes form the parietal layer, and afferent, efferent arterioles. Here are the capillaries of the glomerulus. All right. So essentially, what's going to happen is as that blood comes into the glomerulus, fluid that is able to pass through what are called filtration slits. You can see how these podocytes, these visceral epithelium, has sort of finger-like projections on them, which are called pedicels. They fit together. Those pedicels act like a sieve that is going to filter out fluid from the blood. All right, just water and small solutes are going to be able to fit through that. It's almost as if you were to take, you have some seawater, you reach down into the ocean, and you pick up a bunch of shells and sand, and you pick it up in your hands with your fingers like this. The water is going to run through the fingers, and the shells will be left behind. The shells will be like the red blood cells, and the water passing through your fingers would be the filtrate. That filtrate then passes out through the proximal convoluted tubule at the urinary pole. All right, so here's a similar cartoon, just a little bit different in its depiction. We see we're zooming in now a little bit more on the actual structure of these podocytes. So we have this is a cross section here. We have the epithelial cells of the capillaries coming in. We see them here. And we talked in the cardiovascular chapter how you can have continuous epithelium in a capillary or fenestrated. Fenestra meaning window. And so these are fenestrated ones with pores in them on the epithelium. Those epithelial cells are then coated in these podocytes. So it's a double cellular layer here. And this podocyte, here's one of them, has all these little finger-like projections that extend outward and interlace with the finger-like projections from the next cell. Podo meaning foot, site, cell. It's like these cells that have lots of little feet pedicel, meaning the feet on the little fingers on the foot cell, the podocyte. What else? Oh yeah. So something to point out is this structure here. We're going to talk about it more. It was in the lab outline, although I haven't been able to get the department to buy a model. Next year, I'll get a model that'll show this a little bit better. But this structure here is called the juxtaglomerular complex. Don't get this word confused with juxtamedullary, right? Juxtamedullary refers to a type of nephron with the long loops. Juxtaglomerular means here's the glomerulus, juxtaposed right next to it is this structure, and we call it the juxtaglomerular complex. This thing right here, so this is the proximal convoluted tubule. It's going to go around, go down through the loop, and then come back through this distal convoluted tubule before it heads over to the collecting duct. This swoop back through here, we have three different tubes, all in close proximity. We have the afferent arteriole, the efferent arteriole, and the distal convoluted tubule. This would be a really good regulatory center, right? So where the kidney or the nephron can say, well, what's the blood pressure coming in? What's the blood pressure coming out? And what's this stuff look like? What's our filtration rate downstream? So it can compare these different quantities and affect the functioning of this nephron by constricting, by reducing the flow through here, or maybe reducing the flow through the glomerulus by the action of what are called these mesangial cells. Mesangial cells. Mesangial cells are little cells that insinuate themselves between. They're not really depicted here in this picture, but they are here as shown in this slice, which is taken off here. This mesangial cell is between adjacent branches in the glomerulus. There's lots of them all throughout the glomerulus. They can contract and affect the diameter of these capillaries, thus constricting and reducing flow, or dilating and increasing the blood flow through the glomerulus. That's all under the control of this juxtaglomerular complex. So the juxtaglomerular complex is made of two regions. Basically, there are these juxtaglomerular cells that are sort of wedged between the afferent arteriole and the efferent arteriole. They also wrap around the afferent arteriole. You see this stuff here. This is going to control the diameter there. And then this macula densa, the dense macule right there, can affect the diameter of the distal convoluted tubule. All right. So glomerular filtration. It's a passive process. We're not burning ATP here. We're not burning ATP. Where are we getting the energy for this? It's coming from blood pressure. So for those of you who on the last day come over to my house for dinner, you may want to wash your hands before you eat. You'll go to the sink. You'll turn on the sink. And we have this weird thing that attaches, somebody gave us, it attaches to our spigot, that the water, as it's coming out, spins a little turbine, and then in a light, it lights the spigot up, and that light is a color that indicates the temperature of the water. Right. So where is it getting the energy to light that light up? And it's getting it from the turbine that is being driven by the water pressure coming out of the spigot. It's kind of the same thing. It's this filtration is being driven simply by the blood pressure. That filtration pressure, the pressure to filter fluid out of the blood into the capsular space is about 10 millimeters of mercury. All right. So think to yourself what your mean arterial pressure was. Think back. Your mean arterial pressure was probably like 85, 90, something like that, maybe 75. And what happened? The filtration pressure is only 10 millimeters of mercury. How come we're not, and this is our blood pressure in here, the pressure in here, the map is maybe 60, 70, maybe 80. Where did all that go? How did we get only 10 coming through in the filtration pressure? You want to take a wild stab before I tell you in the next slide? Yeah.
Is there more volume?
What's that?
Is there like a larger volume?
Is there a larger volume over here? It is a larger volume, but this volume is full of fluid. There is fluid in there. But you're on the right track. What about this fluid that's in here is going to affect what's happening? Yeah.
Maybe all solute?
Okay, so there's one thing. She's on the right track. What kind of solute? So first of all, there's solutes that are just passing, the little solutes that are passing. But we have some other solutes here that are not passing, these plasma proteins, right? Those are called colloids because they're suspended. They can be spun down if we did, the centrifugation. These colloids exert their own osmotic pressure, right? Because there's a high concentration of this is a kind of a chemical gradient. There's an osmotic pressure that's driving water back this way. It's the osmotic colloidal pressure. You guys follow that? Yeah. It's because there's none of this protein over here. Because of that, water is going to want to flow back in here to ease this gradient. There's a real colloidal osmotic gradient over here. So that's one thing. What is the second thing? What pressure do you think this is at here? Yeah, it's hard to guess, right? It's hard to guess. You could guess that, say, if your blood pressure, your MAP is at 60, your mean arterial pressure is at 60. If this is at 10, and I told you your colloidal osmotic pressure is at 20, then you could guess what this pressure is here. Couldn't you? So if the colloidal osmotic pressure going that way is 10, the filtration pressure is 20, the filtration pressure going this way is 10, and your mean arterial pressure is 20, it's 60 in here. That leaves 30 left over. You follow me? That means the fluid pressure in here is probably 30 millimeters of mercury in the example I'm giving you. So it's fighting against, your blood pressure is fighting against the fluid pressure in the capsule. It's just like the concept of afterload in your heart. So remember, the ventricle had to fight against the pressure in the aorta before the doors snapped open and it ejected the blood. Right? It was that afterload. It's the blood pressure in the aorta. It's the same thing here. Your blood pressure is fighting against the fluid pressure that's here. So the net filtration pressure is only 10 millimeters of mercury. If the pressure of fluid here was equal to your mean arterial pressure, you wouldn't get any filtration at all. All right. Okay. So looking closely at this membrane, we have the endothelial cells right here with their fenestrations, their little pores. These are the filtration slits of adjacent pedicels, and then stuff just fits right through there. Okay. Here is an SEM of a podocyte as seen from the urinary space. Pretty cool. Look at all those little podocytes interdigitating around the capillaries. All right. So what makes it through? Water, electrolytes like sodium, potassium, calcium, whatever, glucose, amino acids, small fatty acids, vitamins, urea, uric acid, and creatine. These are all things we want to get rid of. These are all things that we probably don't want to get rid of. What doesn't make it? Blood cells, proteins, anything large. So this is pretty much everything that would get spun down into the hematocrit in a blood sample. So here it is. This is what I was talking about. Say the hydrostatic pressure of your blood is 60 millimeters of mercury. All right, pushing outward, out through these filtration slits. The colloid osmotic pressure, that's that osmotic gradient of all of those things that can't fit through the slits that are still in suspension. That's 32 millimeters this way. And then that back pressure of the fluid in the tubes back here in the capsular space, they're putting it at about 20 millimeters of mercury. So that in this example, your net is about 10 millimeters of mercury. This value is modifiable. We can modify that. The juxtaglomerular complex is able to modify that. It's able to modify that by adjusting this number here. Right? This number is pretty fixed based upon the composition of your blood. This number here is a little bit modifiable by the action of the macula densa on the distal convoluted tubule. But this number here we can affect by changing the blood pressure of the blood coming in at this afferent arteriole. We'll see how that works when we talk about the juxtaglomerular complex. Your kidneys are really, really sensitive to blood pressure. So high blood pressure in your glomerulus can make the kidneys to hypertensive destruction. So here is this is nephrosclerosis, hardening and scarring of the kidneys. What happens is if you have really high blood pressure, it's going to rupture this glomerulus. Those cells are going to rupture because you're squeezing fluid through it too hard, and it's just going to burst those really fragile, thin vessels, and it gives you you can get atherosclerosis of hardening of the blood vessels in the kidneys, and your kidneys die. Bad state of affairs. All right. Well, so I've already sort of hinted at it. What are the ways in which we can control this glomerular filtration rate? The glomerular filtration rate is directly related to that net filtration force that we're experiencing in the glomerulus. There's autoregulation that happens right in situ. There's various hormonal mechanisms that we'll talk about that are initiated by the kidneys. That's erythropoietin and renin. And then there can be autonomic regulation via the sympathetic nervous system. All right, so let's take another look at this juxtaglomerular complex. It's an endocrine structure that can secrete these two different compounds, the hormone erythropoietin, and so this hormone gets produced when the oxygen levels in the kidney fall off. So there are chemoreceptors in the juxtaglomerular cells that are monitoring the ambient oxygen levels that are coming into the glomerulus. When that falls off, they're going to produce erythropoietin and secrete it into the blood, where it's going to travel to the spleen and the red marrow for production of red blood cells. Okay, that's going to have two effects. It's going to increase the oxygen carrying capacity, which was the initial variable that was noticed, and it's also going to increase your blood pressure by increasing the hematocrit in your blood volume. The second is the enzyme renin that's going to elevate your blood pressure by a very complicated, we could spend an entire lecture just talking about what renin does, frankly. It goes through a very complicated series of, a very complicated cascade, which I did put up a very general schematic of, I forget what day, the cardiovascular day, I believe, that will ultimately result in an elevation of your blood pressure and an elevation of the glomerular filtration rate. It makes you thirsty. It affects the osmoregulation in the blood. Okay. So if your glomerular filtration rate rises, the flow of your tubular fluid is going to increase, right? So if you're filtering out more fluid is going to be passing through the tube. If that happens, you're going to, the juxtaglomerular complex is going to be stimulated to absorb more sodium chloride, because you don't want to deplete the body of salt too rapidly. The macula densa is going to stimulate those juxtaglomerular cells with various paracrine hormones that are going to act in situ in the locale. It's not endocrine. Remember, paracrine, that was one of the sort of tricky questions that I threw at you on the quiz. One of the options on one of the questions was about paracrine secretion. Paracrine is one cell produces a hormone that affects a cell adjacent to it. So it produces these paracrine hormones that this macula densa produces paracrine hormones that are going to affect these juxtaglomerular cells that you find right here. Those cells can contract and are going to constrict this afferent arteriole right here, thus reducing the blood flow into the glomerulus and thus reducing the glomerular filtration rate. Another impact of the paracrine secretion is to affect the mesangial cells that I said are sort of intercalated here between the capillaries and can contract and constrict the diameter of those capillaries. Also affecting the glomerular filtration rate. On the other hand, if the GFR falls off, that means we're not filtering as much. The macula is going to release, is going to relax the afferent arterioles. So this is going to release their control over the the juxtaglomerular cells here, and we're going to have an increase in blood flow and an increase in filtration. Okay. The increased blood flow is going to bring that filtration rate back up. All right. So here's that same process in this sort of homeostatic cycle depiction that we've used all semester. So in the beginning, homeostasis is disturbed. We have a reduction in the filtration rate, a reduction in the filtrate as sensed by the juxtaglomerular complex in the distal convoluted tubule, and it's also going to lead to just a decrease in urine production. When that happens, we'll have the juxtaglomerular complex is going to dilate the arterioles carrying the blood into the glomerulus, and also going to contract the mesangial cells, so doing increasing the hydrostatic pressure so we're sending more blood in, and then the mesangial cells are putting that blood under more pressure, and so that hydrostatic force, the blood hydrostatic force, is going to increase, and the net pressure is going to go up, and the filtration rate will respond accordingly. Also, we dilate this, so we let more blood in, but we can constrict the amount of blood coming out. So it's feeling pressure on the front end as it's trying to exit the glomerulus. So that's also going to lead to an increase in blood pressure. More blood coming in, less blood coming out. The blood that's in there is under higher pressure. All of that is going to increase the blood pressure in the glomerulus, and that's going to increase the filtration rate. So your glomerular filtration rate is about 125 milliliters a minute. Just to put that into context, Brittany's water bottle there, that little blue water bottle, if she were to fill that up, that would be maybe 400 milliliters, maybe 450 milliliters. So it would take your kidneys 3 minutes to fill that bottle with fluid. Your kidneys are doing that all the time, every day. You would be a dry husk of a human being by the end of class if your kidneys did not put significant effort into reclaiming all of that fluid that it's filtering out, right? That's a huge amount of fluid. That's 180 liters in a day. 180 liters in a day. That's about 50 gallons or so. It's a lot. It's a lot. A 50-gallon barrel your body is filtering out in a day. So 99% of that or more is getting resorbed again. So you're putting all this energy into filtering it out, but then we're going to resorb all of that water, the vast majority of it. So about 10% of the fluid entering the kidneys leaves the bloodstream. The rest of it, about 10% of the fluid entering the kidneys leaves the bloodstream and enters the capsular space. Okay, so even that, at the 50 gallons of filtrate that your glomerulus is doing a day, it's only 10%. So that means there's 500 gallons of blood pumping through your kidneys a day. It's pretty serious. All right. The homeostasis of the filtration rate and the role of renin and the angiotensin system. This system gets really complicated. It gets really complicated. And so this is a sort of stripped down version of what's actually happening here. But we'll try to take baby steps through this. So starting with the normal homeostasis, homeostasis gets disturbed. We have a decrease in the filtration rate. That can be due to to a loss of blood volume for some reason. It can be due to a change in your systemic blood pressure. Maybe you have a renal artery blockage, which is a problem. Anyways, that is going to cause a release of renin by the juxtaglomerular cells. And sympathetic nervous system can also stimulate that release of renin. Renin, we said, is an enzyme. It's going to cleave angiotensinogen to angiotensin. Angiotensin goes through a long cascade, a big molecular cascade that involves multiple organs in the body, which eventually lead to an elevation of blood pressure and an increase in your blood volume. All right, so that's the generalized thing. Here's just a very a little bit more detailed depiction of that. So drop in blood pressure, produce renin. Renin cleaves angiotensinogen to angiotensin 1. This heads to the lungs, where it's converted to a different form of angiotensin. This angiotensin 2 is the one that is broadly active throughout the body. It affects the hypothalamus. It's going to affect the suprarenal cortex, which is going to produce aldosterone and have all the downstream effects of aldosterone. Angiotensin 2 is going to lead to vasoconstriction, which is going to bring your blood pressure up. And the hypothalamus is going to make you thirsty and want to get a drink of water. And all of those things are going to lead to an elevation in your blood pressure. So there's a lot of magic in these arrows and these arrows here. Not magic, but just lots of other steps that are not being depicted there. All right, so the negative feedback control. If you have a high filtration rate, you're going to have a rapid flow of filtrate that's picked up by the distal convoluted tubule and that juxtaglomerular complex. This is going to be sensed by the macula densa in that region. And we're going to have the paracrine secretion, and that's going to constrict those afferent arterioles and reduce the GFR. That is sort of the take-home message of the last several slides, just sort of a review. That's the bottom line, the generalized concept of what's happening there. All right? Okay, so that's how the glomerulus works. We're going to move into the convoluted tubules now. This slide has a lot of details on it, and it's because I wanted some of this, but I didn't want all of it, and I couldn't find an editable version of this slide, so I just put it on there. So I don't need you to know all the details of everything that's happening in here. The important point that I want you to see is that, first of all, the proximal convoluted tubule is responsible for bringing water back, that 99% of the filtrate that we need to reclaim, right? The 50 gallons a day that we're filtering. The proximal convoluted tubule is resorbing that, but it's also resorbing a host of other organic nutrients and ions. So those include glucose, carbonate, and the passage of potassium. All of these processes, everything that's happening in the proximal convoluted tubule is going to, in some way, involve sodium. So sodium is that's kind of the point that I want you to get from this slide, is that sodium is in some way responsible for bringing glucose back. Sodium itself, so you can see it's a co-transporter here. Sodium is used as a co-transporter to pump acid out. It's removing acid, so neutralizing the blood here. It's also, and that is that's part of this carbonate shift here. It's exporting, it's a co-transporter and getting this carbonate out of back into the blood there. It's involved in this potassium transporter. This is our old friend, the potassium-sodium pump, the ATPase. So sodium is essential to driving a lot of these processes. And that should make sense to you. There's a lot of sodium in your blood, and thus there's going to be a lot of sodium that gets filtered out into the tubular fluid. So we might as well use that sodium. Instead of losing it, we're going to use it to drive a lot of this resorption processes. Okay, that's the PCT. That's all I'm going to say about it. Next is the loop of Henle, the nephron loop. And this is a little bit of a nuanced mechanism here. Let's see if we can figure it out together. This is the process of countercurrent multiplication. So the two loops, in general terms, the job of the two loops, the descending loop is to bring water, to continue to reclaim that water as we were doing in the PCT. And then the job of the ascending loop is to reclaim more sodium. We're going to reclaim more sodium. All right. So this descending limb allows water to pass, but no ions can come through here. So this is solute impermeable, and water is just osmosing out of here. All right. This in the beginning, it's a very low concentration, 300 milliosmolals per liter. That's a concentration, and the ambient concentration out here is going to match that. As we pass down, this water is going to want to osmose out into it's going to want to osmose out into the ambient environment. Why? Well, yeah. So this is an active, any kind of solute pumping in. So this concentration gradient. Let me get this straight. The concentration gradient that's being established along that descending limb is helping to drive the other process that we have on this side, right? So this is causing a concentration gradient that is being reinforced by the efflux of sodium ions on the other side. So here we have 100 milliosmolal, and this ambient concentration is at 300. So that's going to want the sodium's going to want to diffuse out of this region, out of the tubular fluid into the extracellular space surrounding that tube. Yeah, so it is this increasing solute concentration as we go down, because we've been removing water, which is helping to drive this solute removal on this side. Does that make sense? Did I muddle that with my sentences? It's going through. Okay. So a huge amount of water is removed here. A huge amount of water. About 80% of the water that enters the descending loop is removed by the time it gets to the distal convoluted tubule. So this is a major location for the resorption of water. All right. Next, the distal convoluted tubule. The distal convoluted tubule, as I had said, is it does two things. It continues the resorption of water. It also has the selective, fine-tunes the composition of the tubular fluid by either secreting toxins and drugs or the selective resorption of ions and nutrients. This is under the control of antidiuretic hormone. So without antidiuretic hormone, the DCT is not going to resorb a whole lot of fluid, and we're going to have a large volume of dilute urine. When we add antidiuretic hormone, this distal convoluted tubule and the collecting duct is all going to suck up a lot of water and give you a much smaller volume of concentrated urine. So throughout the entire length here, we're pulling in water here, a huge volume of water through the loop of Henle, and then we continue all the way down. So throughout the entire length of the nephron and the collecting duct, we're able to concentrate the urine and reserve water. The biggest place for modification of how much we concentrate the urine, how much we reserve the water, is here in the DCT and the collecting duct under the control of antidiuretic hormone. And this is being produced by the endocrine system under the control of the hypothalamus and the pituitary. All right. Here's an important idea. The ideas of transport maximum and renal threshold. This picture is in your Netter notebook. So the transport maximum, the definition here, maximal rate of secretion or resorption of a substance by the renal tubules. This means that it's like you have a building and there's a revolving door in the building, and we're all going to go into the building, or like a turnstile, and everybody wants to go into the stadium to watch a game. And in the beginning, it's fine. Everybody's passing through, and nobody's having to wait in line. But as more and more people build up, those turnstiles get saturated, and only so many people can enter the stadium, and you sort of reach a max capacity. And it doesn't matter if you have a zillion people behind you. There's only so many that are going to be able to get past the turnstiles at a given time, at a given rate. That would be the transport maximum in this example. So here we have some kind of transporter that is pulling in, say, glucose. This is a glucose transporter driven by the sodium that I showed you in that one slide. And right now this transporter is pulling in glucose, and it's not saturated. We could pull in more. There are spaces open on the pinwheel here as it's pulling glucose out of the filtrate that we've lost when the blood got filtered, and it's throwing it back into the blood. Here, this is at the transport maximum. So here's the concentration of the whatever that is, maybe glucose, as it's going down the proximal convoluted tubule. It is getting transported across this transporter into the blood, but it's at its maximal rate. There's no open sites. And then if we have a really high concentration of whatever this thing is, it sort of swamps it out. This is moving as fast as it can, but someone's going to pass through and enter the urine. All right, so this is in resorption. The same thing happens with secretion. Maybe we're trying to get rid of something that didn't pass out in the filtrate, like some kind of toxin or whatever that didn't pass through, and it's being actively secreted. You can have here's the transporter below its transport maximum. Here, all the sites are active, and here it's working as fast as it can, but it's not removing everything from the blood, and some is passing the capillaries. So The renal threshold. That's the concentration in your blood at which a specific compound or ion is going to begin to turn up in the urine. All right, so what is the concentration of that stuff right here in the blood where we begin to see it appearing in the urine? This is going to depend from compound to compound based upon the identity of the transporter that is being used to move that solute. So glucose has a pretty high renal threshold. If you have 180 milligrams per deciliter, that you're at your transport maximum and the renal threshold. If you go above that, you have glycosuria. That means glucose in your urine, glycosuria. So if you have higher than 180 milligrams per deciliter blood sugar, you may have diabetes, like your body's not able to keep up the insulin in response to the blood glucose. They call this diabetes mellitus. Diabetes refers to the condition where you're urinating a lot. People with diabetes mellitus urinate a lot. Mellitus means sweet. So diabetes mellitus is in contrast to diabetes insipidus. This is because, what was the guy's name? Hippocrates used to take a little, he was looking at the bubbles. Well, that wasn't the only thing he was doing. They would taste the urine, and if your urine was sweet, they called it diabetes mellitus, or sweet urinating disease. And that was indicative of this problem of high blood sugar. Yeah, you guys are thinking it's gross back there. It's sterile. Your urine should be sterile. Never drink it. But and diabetes insipidus is a different disease where there is not actually glucose in your urine. It's not related to blood sugar problems. It's a totally different disease. So amino acids have a much lower renal threshold, 65 milligrams per deciliter. It's actually pretty common to see amino acids in your meal if you're like the paleo diet kind of person, where you're eating lots of meat or beans and nothing else. It's called amino acid urea, and we'll talk about nitrogen balance in a little bit and what that means. The word natriuresis is the secretion of urine, sodium in your urine. We talked a little bit in the endocrine chapter, not a very little bit. I think I mentioned the natriuretic hormones that are produced by the heart, the endocrine function of the heart. Those are hormones that can affect this process here, the secretion of urine of sodium into the urine. Diabetes mellitus. So there is two types, type 1 and type 2. Chances are high that everyone in the class has somebody in their family or knows somebody that has type 2 diabetes. Type 1 is juvenile diabetes. This is early onset. It just is a problem with your pancreas. Your pancreas does not produce insulin. That's only about 10% of the diabetes mellitus that you're going to run into out there. The vast majority of people with diabetes mellitus is insulin resistance, adult onset diabetes. People with this type of diabetes have a much higher than normal risk of heart disease and stroke. They have 20 times the risk of limb amputations due to peripheral vascular disease and gangrene. It's also the major cause of diabetic retinopathy. retinopathy, or the death of the retina, and blindness. Non-traumatic blindness. It's also the largest cause of kidney failure. It can lead to these other things: dementia, sexual dysfunction, and it can reduce your life expectancy by 10 years. Tomorrow and on Monday, in that talk from the guy, I forget his name, but we'll talk about metabolic disorder. Metabolic syndrome, diabetes mellitus, adult onset, is one in a constellation of different disorders that's part of metabolic syndrome that probably most of you have already heard of a little bit, but we're going to talk about it more. There's this thing called gestational diabetes. This is for people who have, this is for pregnant women who get a transient diabetes during their pregnancy. About 3 to 10% of women go through this process. This does slightly increase your likelihood of having adult onset diabetes afterwards, but typically the diabetes resolves after postpartum, after the baby is born. So, so here's just a constellation of different symptoms that you may see with a person who is diabetic. Some of the interesting ones on here you'll see is this the smell of acetone. What's interesting is that what's happening here, these people who have diabetes mellitus, even though their blood is literally awash in glucose, there is so much glucose that they're urinating it out, right? They have lots of glucose. Their cells are insulin resistant, and they are not able to pull that glucose into their cells. So they are literally starving to death, even awash in all the nutrients that they need. And their brain needs that glucose to function. Well, there is an alternate pathway whereby the brain can metabolize these things called ketone bodies. And without going into that in detail, these ketone bodies end up in the bloodstream and give people with acute uncontrolled diabetes mellitus this smell on their breath that is reminiscent of nail polish remover. The other category of person that you're going to see, maybe, say you're in an ER or something and you see one of these people, the other category of people that has the same pathology or same presentation is alcoholics, like really severe alcoholics who are their problem is they're not eating anything else. They're getting all their calories from alcohol, and their metabolism is doing the same sort of thing. They're producing ketone bodies, and it gives their breath that characteristic smell of acetone. What else is interesting in those? Well, anyways, this here is the symbol. You ever see somebody with a wristband, one of those medical wristbands that has this blue circle on it? That means they have diabetes. That's the symbol, the international symbol for diabetes mellitus. All right, type 2. This is, again, I need to next year. I'll do it next year. I'll update some of these statistics. They're a couple years old. But here is the American Diabetes Association and the World Health Organization data that I collected all together. There are about 25.8 million. This was in 2010, so this is 5 years ago, and these numbers have been going up. There's 25.8 million children and adults in the United States. That year, it was 8.3% of the population who have diabetes. That is an epic number. That is a pandemic. That's a pandemic. right? I mean, I don't know if anybody, and I'm not going to poll whether anybody has it in this class, but that would mean that at least 2 people in this class would have diabetes mellitus. Now, that's probably not the case because diabetes mellitus has a very strong lifestyle correlation, at least type 2 adult onset. A large number of these are undiagnosed, so the lack of the actual clinical presentation, the diagnosis rate of this is only about 60%. So of that 25 million, only about 19 million have been diagnosed, 7 million have not. So these are the people with it. Here are the pre-diabetics. These are people on course to at some point in their life develop it. 79 million people. 79 million people in the United States. There's only 315 million people in the country. That is gnarly. That is really a major, major problem. This is why having regular checkups with the appropriate blood panel, glucose challenges, all that stuff is really important. So there were 1.9 or almost 2 million new cases diagnosed in people your age or older in 2010. This is not just an American problem. America is exporting its diet. In 1985, there were only 30 million people globally. In 2010, there are now today there is certainly above 300 million people with diabetes in the world. Let's look where that falls. A lot of them right here in Europe. It tends to focus, and in this part of the world, these people certainly have a sweet tooth. I don't know. I know Indian culture and Saudi culture, Turkish culture. These are all cultures that eat a lot of sweets. Certainly Australia, New Zealand. So you can see where a lot of it is concentrated. It's the export of the American diet. The world average is 28.3%. It's really bad. Oh, wait, one number. There's a number in there I wanted to point out. Oh, yeah, here it is. 218 billion, probably closer to $250 billion right now. Yeah. The U.S. GDP is only about $17 trillion. Okay. So this represents a really sizable fraction of the U.S. GDP. Summary. We have glomerular filtration of the blood, which is controlled by the juxtaglomerular complex. The filtration rate is controlled by this juxtaglomerular complex. We have the filtrate, which then enters the PCT. There's a bunch of nutrients that are pulled back into the bloodstream, primarily in tandem with sodium. And a lot of water is pulled back in here. We have water, the osmotic gradient and the resorption of water that creates this osmotic gradient, which then helps to drive this sodium resorption on the ascending loop. Distal convoluted tubule, we have variable resorption of water under the control of antidiuretic hormone and the active secretion of other toxins and electrolytes that we may want to remove from the body. This tubular fluid ends up in the collecting ducts where this process started in the DCT continues until it passes out into the calyces. All right. So, and this is just a different way of looking at that based upon the different kinds of things that are being secreted. We have, this is the percent remaining in the filtrate. So the filtrate we have here, the creatine actually goes up because we actively secrete creatine in the proximal convoluted tubule, and it stays constant throughout. Here is urea. We filtrate quite a bit of this out, and then some of that gets resorbed by the PCT, gets re-excreted in the nephron loop, and then gets partially resorbed. Ending at about 60% of what the filtrate was. The glucose is pretty much, as long as we're below transport max, glucose is completely, and the amino acids are all completely resorbed. And the black line is the total osmolarity. And the sodium here we see is the ion that's overwhelming pretty much everything else in terms of the osmolarity of the tubular fluid. And then this is the amount of water that's being released without the presence of ADH and the further resorption in the distal convoluted tubule. If you do have ADH, then this number goes down quite a bit, and we resorb the last of the water for very concentrated urine. All right. A couple more slides. We're going to end early today. Micturition is the word. It's another medical term you'll come into contact with for the process of urination. This micturition is simply going to the bathroom, removing urine from the urinary bladder. Diuresis. Diuresis is not just micturition. Diuresis is the entire process of removing volume, water volume in the body. So maybe you have a lot of extracellular fluid that is wherever. Diuresis would be pulling that fluid out of the interstitial spaces into the blood and then filtrating it out and then removing it from the body via your urine. So that's diuresis. A diuretic is a compound whose job it is to stimulate that process of diuresis. So it's not just encouraging urination, it is encouraging that entire process of pulling fluid out of the interstitial spaces and putting it into the blood for then filtration into the urine and removal from the body that way. So diuretics are going to reduce extracellular blood volume, your blood pressure, and all three of those things. There's a couple different types of diuretics. I put them on there not because I need you to know them at all, except to point out that they have different places in different diuretics are approaching the problem from different points in the system. You can have loop diuretics that are affecting the function of the loop of Henle. You can have ones that are affecting the distal convoluted tubule, aldosterone agonists there with spironolactone. Okay, so a diuretic would be a kind of compound that you may apply to a person who's in congestive heart failure, for example. Is a person who has a lot of fluid in their pericardial cavity, and so a lot of fluid pressing on the heart, preventing perfusion of that heart via the coronary vessels. You need to pull fluid off. You give them a diuretic, they urinate all that fluid out and relieving pressure on their heart. All right. The nitrogenous wastes. So here are some of the main nitrogenous wastes that your body would be removing. They come from two different sources, either proteins or nucleic acids. So proteins get broken down to amino acids. They then get broken down to ammonia, which gets converted to a little bit more soluble form of urea in the liver. And then nucleic acids. Uric acid, you see right there, is from nucleic acid catabolism. Uric acid can then be uric acid, and then also creatine is another catabolic breakdown product of nucleic acids. And creatine phosphate breakdown. So sometimes we are going to remove high levels of creatine. So when we look at the composition of a standard sample of urine through urinalysis, these are what you will see. The pH of your urine is typically more acidic than your blood, so it is one way of alkalizing your body by removing acid from the body. The pH ranges from 4 to 8, but hovers around 6. It has, what else is interesting there? You urinate anywhere from 700 to 2 liters, milliliters a day, 700 milliliters to 2 liters a day. That should be about the fluid intake that you should have too, depending on the specifics. It is sterile, as I said earlier. Thank Hippocrates' lucky stars. What else is interesting in this list? So, what we can see here that's interesting is we tend to remove potassium from the body in the urine. We also tend to lose a little bit of sodium in the urine. But right here, we can see, this is sort of interesting, that glucose, the plasma level of glucose is far above that of urine. Usually hardly any glucose, unless you have type 2 diabetes, is getting into the urine. Same with amino acids, unless you're paleo, as I said. There should be no actual protein in the urine. If you have protein in your urine, that is indicative of kidney damage of some sort. And then, yeah, these numbers here, we can see there's the active removal of urea and creatine. So these concentrations are far higher. These concentrations are far higher than what you'll see in your actual blood. Which means those have to be removed actively by the various convoluted tubules, particularly the distal convoluted tubule. All right, almost there. So pylograms. These are. This is a form of imaging that is used to identify kidney stones. Has anybody had a kidney stone before? Good. The way they do this is they catheterize you, they stick a contrast, a dye, into the catheter, travels up the urethra, and helps you look at the structure of the urinary tract. So here we have the bladder, the urethra, here's the hip, the major calyces, the minor calyces. Both sides, this person looks pretty good. Here's another not colorized one. This looks pretty good all through here. Happy flow. We have bladder, happy flow here. Ow! Oh! There's a kidney stone right there, and we don't have any flow on this side. That doesn't feel good. Those are really quite painful. There are a whole host. So not all kidney stones are the same. Here are some examples. They can be crystals of uric acid. They can be a struvite. which is different than uric acid crystals. Here it is ammonia containing, also has magnesium phosphate in it. They can be calcium containing. Whatever they are, they are inorganic solids, or I guess uric acid is an organic compound, but they are crystalline solids that have low solubility. They don't stay in solution very well, and they can also indicate metabolic disorder of one sort or another. Anyways, the appearance of any of those can really be a problem. So here we have the kidney stone. Here you can see there's some real problems in that kidney right there. It's getting distension of the calyces. So this must have been a systemically applied contrast. So these calyces are largely distended. This person can't urinate, whereas they're coming down here. So this kidney stone is blocking that ureter. You can see maybe it's sort of tortuous through here. Sometimes they just pass. Sometimes they do ultrasound, break them apart. I don't actually know a whole lot about the removal process beyond that. So 80% of kidney stones happen in men. All right. Renal insufficiency, hemodialysis. For people whose kidneys just stop working, there's this process whereby you hit an artery, you pump the blood out, it goes into this machine where the blood runs through this dialysis tubing that's sitting in a hypotonic solution, and they try to create a solution where the things that you would normally resorb, like your glucose and whatever, those are isotonic but hypotonic for the kinds of solutes that you want to pull out of the blood. All right, so that's the dialysis fluid with the dialysis tubing. Then you just pump back into the body. Here's a bubble trap to make sure you're not getting any embolisms into the body. This works, but not especially well. This guy's not going to be feeling the tiger in his tank for a couple days afterwards. So, I think that's it. Are there any questions?